Hello again, everybody. It's time for Q&A with A&V. I'm Vincent Racaniello, and with me is Amy Rosenfeld. Hello, Amy.
Hello, Vincent. Did you have a good day today?
Yeah, I saw you today in the lab.
Yes,
Hey,
I
folks,
did.
this is cool. Amy brought me to see space in New York to have a studio for microwave TV, right?
Yep. We've whittled it down to one spot. It's almost time to sign the lease. That's the next step.
Yeah, so when the pandemic is over, we can have a studio and you folks can visit.
Yes. Go visit him. Don't come visit me. Visit him.
They don't want visitors?
The lab is a dangerous place. It's when you drop CO2 tanks on your foot and stuff, it's not good. It's too
So
dangerous.
we got somebody from Colorado. Hello, everybody. Wisconsin. We're in
I prefer the
Manhattan.
person from Lebanon.
The studio is going to be right next to Madison Square Garden in One Penn, right? That's the address.
Yes. One pen. 24th floor.
Kosovo. Good job, Doctor.
I think
Germany.
the Rima is from Lebanon. I think that's great.
So I found this app, which I will use next time, which shows a globe where everybody is from in the chat. How cool. That's what I've been looking for. New
Okay.
Jersey. Questella wants to know what's your favorite wine?
Me or you?
You, Amy.
Oh, I like bubbles. So I like champagne, prosecco, cava. I like bubbles.
Upper West Side, Chicago. That's Amy's former stomping ground. Colorado again.
Yeah, the West Loop is good.
Don't forget to
There's a
give
lot of
us
good restaurants in the West.
Asheville, North Carolina, Ohio. Don't San Diego. Don't forget to give us a thumbs up down below. Just in Ohio. All right,
All right,
Texas,
let's
Oklahoma.
move on. I have to do plaque assays. My
Thanks.
overlay.
Oh, here we go. Brazil. Very good. All right, so I
Chile, you
saw.
missed the person from Chile. I mean, come on.
I'm looking for the first question here. This one is a good one. It's from Melissa. I've heard many people say they're wary of the vaccine because there's no way to know long-term side effects. What is your answer? I have my first dose tomorrow.
Roll up your sleeve and get a vaccine, put a cute band-aid on it, and go back in 4 weeks.
I would say that any of the frequent. Well, I would say that we've been a couple of months now. Haven't seen anything very serious. There's possibility long term, but they would be rare. And I think COVID is more dangerous than that would be. So I think that you ought to get the vaccine. Trinidad and Tobago. Cool. That's very cool. Arctic Circle. No, Norway. Okay. Would you have Dr. William Hazeltine as a guest?
No.
Nope. He said too many silly things. He's not thinking clearly. Um, don't worry, be happy says. Do folks who have already been exposed to SARS-CoV-2 need only one shot?
Well,
No.
the protocol is 2 shots unless you're doing Johnson and Johnson. And so since it's not a question on the questionnaire, you're going to get 2 shots.
Yeah, and we don't know what your immunity is after natural infection. So we want to make sure that you get the vaccine. Hello, Andrew from New Zealand. Kia ora. Cool. Oh, this is for Amy. This is from Raphael. I feel the most important answer isn't in any paper about vaccine efficiency. What is the level of T cells? Variants will always mess up antibodies, but probably not T cells. You have any
Well,
thoughts on?
I don't think you're supposed to hold a vial of serum from a vaccinated person up to your head and ask what the T cells are thinking. So when people start doing T cell experiments, we'll know.
So you're right,
Um,
Raphael.
but
People aren't
but
looking
I
at that.
but I do believe that some of the epitopes are T cell epitopes. like
Yeah,
that
and
they can change.
they can change
But
as well.
yeah,
Absolutely right. But we don't know the contribution of T cells.
well, we don't even know if these are in T cell epitopes. If there's like that very little low percentage chance that an epitope of a B cell would overlap with a T cell epitope. You always
Yeah.
have that, minor probability. But as I said, it's been frowned upon to just talk to the T cells. They're very hard to talk to.
It's hard to do T cell epitope assays, but they do vary. Amy is absolutely right. Rima says, "What does B117 mean?" It's just the name of the variant lineage. That one is in the UK, and it's these lineages that have a lot of mutations, and we don't know what they mean. And the others have a B number, like South Africa has, I think, B1.135, and etc. It's just the name of the variants.
I think it refers to a part of where it is located on the phylogenetic tree, I think.
Could be, yeah.
I think that's usually how they do it.
Doug wants to know of what utility to immunity are antibodies to viral proteins presented at the surface of host cells but not on the surface of virions?
What?
So I'm not sure what you're exactly referring to. So the surface of the cell is presenting the antigen to the immune system so that you can have an immune response. And then you make antibodies or T cells to that antigen. And now if the antibody reacts with a viral protein that's not on the surface of the particle, let's say only in infected cells, there are some ways that could block infection post-entry, but most of the antibodies we're talking about with spike, of course, are blocking or attaching to particles and blocking entry. So, Amy, I'm thinking about the antibodies to adenovirus that actually get in the cell and they block infection after uptake.
No, I know what you're thinking about. I just don't understand the question.
Yeah.
So,
Well,
so like the viral proteins on the surface of the virion are not recognized. They're not presented to make immune response. The particle has to be engulfed and digested and stuff, right? So I'm a little confused about the question.
Nazareth wants to know why we're dodging the long-term side effect question.
Because a long-term side effect means that there has been you've waited a sufficient length of time after people got a vaccine. The majority of people started getting vaccinated at the beginning of January. This is February 3rd. This is not long-term. Come back to me in a year and then we'll talk about long-term. So you can't predict.
Yeah, we're not dodging.
It's not a long-term. Right.
We're being honest. We don't have the data. And I just
Well, there is no data. There is no data because long-term implies a span of time, right? Yeah. The majority of people are, 6 weeks at max?
Yeah.
It's nothing.
All right. Bradley's doctor recommended famotidine for recovery.
No.
Now, Daniel said it's not really helping with COVID, right?
It does not help. Somebody here at Columbia, when they used to do the weekly symposiums, it was looking at it and it had no effect whatsoever.
Okay.
It's like ridiculous.
Susan wants to know if there are any concerns with ADE, with adenovirus-based vaccines that are given twice. Do you have an opinion?
I have not seen any indication that there is ADE with the adenovirus vaccines that have been given twice for this virus, in this case. But previous years, like when they did it in the 90s at Duke, there was, but they have addressed those concerns.
Frank wants to know, his sister got the second dose yesterday and got itchy. It's fine. Yeah, that's a good response. It's
neutrophils.
Yeah, is that right?
Yeah, it's neutrophils.
Yeah,
and TLR4.
So it depends on. Go ahead,
yeah,
sorry.
But it ends up being neutrophils and TLR4. For like liver disease, itch correlates with it, and that correlates with TLR4 activation. And then there's been some papers that talk about neutrophils.
Got it. Carlita wants to know if we know any examples of vaccines that have provided more robust, longer-lasting immunity than infection. I'm not sure I understand. Immunity to disease, do you think, Amy?
Sure, or just like if you got disease and your memory B cells disappeared after a while, and then you got a vaccine against them, how long would the memory B cells be present longer? I don't really know that. I don't know why I would ever think that a vaccine is going to give you longer immunity than natural infection.
Okay.
What do you think?
Well, I don't quite understand, but my interpretation is that she wants to know, can you get immunity to disease but not infection? And so my
Can
example
you
is
get
always,
do you know of any examples of vaccines that provide more robust, longer immunity than if you had natural infection?
oh, this is natural infection. I see.
Right. So I
Um,
don't know of any time when you would have a vaccine would induce a longer term immunity than than natural infection would. I don't understand why that would be possible.
Fairy Tales wants to know, a lot of people can't get vaccines because countries are refusing to lift patents at the WTO. Now, do you know anything about this, Amy?
I'm not a lawyer.
There are a number of organizations that are trying to distribute vaccines, and I think they are going to get around that in some way.
So someone said measles vaccine induces better immunity than infection. Is that true?
Natural infection gives you immunity that lasts 40 to 50 years. That's probably comparable to the vaccine. I wouldn't say the vaccine is better.
Well, from my experience, the vaccine did not give me immunity to 40 or 50 years.
No? How do you know that?
Because I got vaccinated at 18 months. I got vaccinated at 5 years before I went to college. I got vaccinated when I went to college. I got vaccinated when I went to graduate school. Then they did a titer, and there was no antibodies.
Here's a good one. Pinky wants to know, what's the likelihood of a true phenotypic change versus just the nucleotides? So in the trade, we call this non-synonymous versus synonymous mutations, right, Amy?
Right.
So how many codons, what's the degeneracy of the codon? Can you figure it out?
There's a part, I think it's 60, and there's 20 amino acids, but 3 of them are stop codons. And some amino acids only have one codon, and other amino acids have multiple codons. It also depends on where the change is, right?
Yeah, right.
So you have 2/3 of the codon cannot be changed, otherwise you will get a non-synonymous change, right?
Yeah. First
And
two bases, right?
and the wobble position can be changed. So 1 out of 3 should lead to a synonymous alteration.
Victor wants to know, why is there a minimum number of particles for infection? Is this to have one successful one that reproduces, or do you need multiple ones to overwhelm defenses? So I think it's both. In an animal, you need to get enough particles. One is enough in cells, but one can initiate infection in cells, but you typically need more because there are a lot of defective particles. And then, yes, in a host, you have the immune system on top of it. So you need more to overcome that because 1 or 2 is not enough. Does that work for you, Amy?
Yeah. I was thinking FMDV, the PFU to defective particle ratio is like 1.
Was that
And
right?
for
It's one? Wow.
yeah,
For polio, it's 200, right?
300.
What is
We
your
wrote it.
virus?
Nobody has done it yet. We're in the, well, one day we will do it, but I haven't done it yet.
Kang wants to know, how many mutations will it take to render PCR ineffective? If you're doing a diagnostic test.
So actually, Nisha and I just recently had that conversation. So you really only need the last 6 oligos of the primer to anneal in order to extend. And
Hmm.
so you probably need about 4 alterations with the rate of the fidelity of the polymerases today. Years ago, you only needed 1, but today
Hmm.
you need more.
Strasbourg. Hello, Gopal from Strasbourg. It's late. Amy, does J&J shot come with a little baby powder?
Probably not, since they're being sued for baby powder.
Really?
Yeah, it correlates with ovarian cancer or something or other, some form of cancer. But yeah, so they're being sued because it used to have talc in it.
Should the J&J vaccine be for people less than 65? I don't see why not. They tested it in people less than 65, right, Amy? So why would it not be? Do you know?
I don't know. I thought that they did.
Oh, look, we have someone from Russia. 4 a.m.
Cool.
Oh, good for you to join us. Thank you so much. Thank you all. Interesting. If the second shot caused a worse reaction than the first, would subsequent booster shots be even worse? Why would you get a third shot though? Right?
Well, if you need to, if your vaccine changes, or if your immunity wanes in 5 years, you need a booster.
Yeah, okay. But I don't think it's going to be worse because if it's years down the line, right? I don't think it's going to.
I have no idea. I have no gauge for that.
I mean, if you take
Yeah.
flu shots every year, you don't get worse reactions every year, right? And that you get
Well,
it.
I think some years, you do get, some years you're unaffected, and some years you're more
Hmm.
affected by the. So I think it's all different.
Yeah. So this year I had a sore arm, and last year I didn't.
Well, yeah, I mean that, but there's a lot of things that go into it. Like some people, when I had a sore arm, like I told my mother, and she said it's because the guy was too hard on your arm. It's like, but
Yes.
then, yeah, so then I referred to it as harpooning. And you
Hmm.
said to me, "What is harpooning?" And I was like, "Really? You couldn't figure out what harpooning was? Remember this conversation about 3 weeks ago?
I do. I do.
I
It's
was
always
harpooned."
you're harpooned.
Yes, I was harpooned.
What's your thoughts on early treatments? Azithromycin, zinc, vitamin C and D, and $1 rapid antigen test strips.
I never like giving antibiotics, so I would get rid of erythromycin or whatever it is. I don't know what the purpose of zinc is, but people swear by zinc lozenges. I don't really see it. Vitamin C and vitamin D, sure, we can go with Linus Pauling, and vitamin D for Daniel. Sure, I'm fine with that. $1 rapid antigen test, never happening. Was a hoax to
But it's
begin
a good
with.
it's a good idea though, right?
No, they should be free because we did the math, and then you were all appalled by the math that I did in October. And then somebody then said, "Well, I was talking to a school and I suggested that they could have a $10 test." And I was like flabbergasted. Like, seriously? Are you kidding me?
How long does immunity last after vaccination, and how long does it last after natural infection? Is there a difference, and why so? I presume this is a general question, not just COVID vaccines, right, Amy?
How do I know? I don't know what they're thinking, but you should take this question because I've already answered it. And
Yeah, so
it's
as Amy said earlier, natural infection tends to provide the longest term immunity because it's a natural infection. It's pathogenic, unfortunately, and if you survive, you've had a lot of virus in you and your immune response has been robust. And you think about a vaccine, if it's a non replicating vaccine, well, it's not replicating, so there's only so much that can do. And the attenuated vaccines are weakened, so they don't make you sick, but they tend to have a slightly less immune response. And what's going to happen with COVID, we don't know. We'll have to see.
well, I also think that I never understood Daniel when he said that that vaccine would induce a greater immunity
I
and
know.
then stuff. I never understood that because there's a lot to immunity besides neutralizing antibodies. And there's a lot of epitopes in nucleocapsid, in ORF8, and
Yeah.
various other proteins that are
Right.
absent when you just have a pure spike based
Yeah.
vaccine. So I don't fully understand how anybody made that conclusion, but I'm not willing to argue with him because I like him.
I'm going to have Paul Offit on Twiv in a couple of weeks. We'll ask him. He's a vaccine expert. All right, but Moderna and BioNTech replaced uridine with 1-methyl pseudouridine in the mRNA to avoid adverse responses. What do you think happens to these modified nucleosides? I don't know what they do. I thought they were to give stability, Amy. What are they for? Do you know the modification, what it's for?
I thought it was stability.
Yeah, so I don't know anything about adverse responses and why that would have anything to do with it. And there's a question coming up saying there's another vaccine from CureVac doesn't have the uridine replacement. Well, it's the same person, JM. So I don't think the uridine, the modification has anything to do with adverse effects. I think it has to do with stability of the RNA. Can mRNA vaccines be given orally? Why not? No, they can't. Because
RNA gets digested.
yeah, the
The RNA would break
nanoparticles
down.
would break down in your stomach, and
Break down in your mouth. You have lipases in saliva.
not me.
Okay.
Just
Well,
kidding.
everybody else but the Racaniello family has lipases in their saliva.
Oh, here's an interesting one for which I don't have an answer. What's your explanation for the disappearance of flu this season?
Vaccination and physical distancing. Not vaccination, masking and physical distancing. It's very clear.
It's not the only respiratory infection that's gone away. Amy's virus went away too.
My
Right?
virus, rhinovirus, adenovirus. Yeah. Tons of viruses went away. I mean, norovirus went away. Everything but a tick-borne virus went away because we all went running around in the country in the summer.
Why doesn't Pfizer or Moderna do transmission studies? Well, Moderna had one planned, and then the funding fell through for it. You got to pay for these clinical trials. And so they're not doing it. I don't know about Pfizer. There's some preliminary. Is the AstraZeneca, was that preliminary data on transmission or just PCR positivity? Do you remember, Amy?
No, it was on transmission.
They seem to see a little effect.
Yeah, it's like half.
50% decrease in transmission. Yeah.
Yeah. And then there's a study from Israel that is on bioarchive that also talks about like a 50% decrease in transmission.
Yeah. So the idea of the single dose, the problem is that there are some interesting results with a single dose, but the data aren't sufficient to allow the FDA to license it. That's the problem. And I'm not sure we're going to get a clinical trial with one dose. Vancouver.
Well, not now that Moderna is like repackaging its vaccine its vaccine vials to have even more doses in it.
Did you catch the Friday Bill Maher segment on lab leak hypothesis? So I didn't see it, but many people have emailed me about this. And
We're
so
still discussing it?
Yes, we are still discussing this.
It's 14 months later. Seriously, there are other things in the world to discuss.
Well, the Emerald X, did you see there were evolutionary biologists on there who didn't give good responses? I didn't see it. I don't know how to talk to people I know in real life. They all think the vaccine is BS. Tests are fake. The virus is just flu, and I'm a crazy person for being so obsessed.
So I read this article in the Times that you should ask them why about how to reason with irrational people. And he talks about like his friend is anti-vaxxer. and he wants to know like why he wants to convince him to be a vaccine person. So he used to like browbeat him and be like, "You're just wrong, blah blah blah." And so then he learned that he read some book or something or other. And he says, you should actually sit there and try and ask them questions. You should interview them about if they can justify their rationale about why they're anti vaxxers or why they think the vaccine is BS or fake or whatever. And then slowly, as they talk, they will either hear where their logic is wrong, or you can, you know, understand why they feel this way, and then you can modify your approach to convincing them. It's a whole editorial in the Times. It was interesting.
Requires some patience, doesn't it, Amy?
Yeah, it's not for me. Like, I don't want to, like, I'm not going to be the person who, like, is going to have these conversations. I'm the person who's going to walk away from these conversations going, oh my God, kill us all.
I think some vaccine hesitancy comes from a memory in older people from the 1976 H1N1 vaccine where people got Guillain-Barré. Yes. So that's true. That was a shit show, right? As Amy would call it, because we didn't even need that vaccine, and it gave a lot of people, about 400 people in the U.S., Guillain-Barré. So
yes, but it was from your university,
yeah,
your department.
However, we don't see that with these vaccines, so there's no Guillain-Barré here, and I understand, but COVID is worse. Let's see. New data from AstraZeneca. Efficacy 6 weeks prime booster interval 4%. Efficacy 12 weeks 82. Well, Amy would say you got to wait longer after that first shot, right, Amy? Not just 4 weeks.
Yes, isn't that what I said to Alan on Friday when I said, "Oh my God, really? The immune system takes time? No way." Really? Never would have guessed.
It takes time, and 4 weeks is rushing it because we want to get through these trials, but ideally you would go longer.
For sure.
Amy Gopal wants to know, and he's asking you, can the long-term effects of COVID be due to latent or suppressed infections, not clearing the virus totally, persistent infections depending on a person's immunity?
Can the long-term effects? Well, I mean, it's an RNA virus. It doesn't usually go latent. It would be hard, and it's lytic, so I don't think it's due to latency. Suppressed infections. What is a suppressed infection? I don't really know what that is. Do you? Is that like chronic?
Yeah, you still have some virus around, and then it recrudesces. But many of the long COVID patients don't have virus; they're PCR negative, so I don't think that's it.
Not only that, but a lot of the long-term COVID actually comes from patients who are asymptomatic and had very mild disease, and then they come back like 4 weeks later with these things, and they're PCR negative, but they've seroconverted. But so it seems that it's way more a consequence of immune dysregulation, and this dysregulation we know that immune dysregulation has lots of side effects long term. So, for instance, a lot of immune dysregulation, or one form of immune dysregulation, is atherosclerosis. Is the overactivation of macrophages and then the atherosclerosis. That's what Aretabe studies.
Dave says, I heard Great Britain extended the time between doses with the logic that the testing was compressed to get vaccines approved. Yeah, I think so. I agree. I think you needed to do a 3-4 week turnaround for the second dose. Otherwise, it would take you a long time to do your trial. I think the only downside, so there's no downside with respect to immunity. I think Amy would agree if you make the booster farther out, but then you run the chance of perhaps getting infected. So I don't know if that's an issue. What do you think, Amy?
I don't think it's the issue of getting infected. I've argued that numerous times over the weeks. It's not an issue of getting infected. It's the issue of people don't want to come back. And I believe that I thought I read that part of the reason why Great Britain was extending the time was not because the testing between doses and immunity was compressed. It was because they wanted more people vaccinated.
Yeah, that's
So they
absolutely right.
expanded it.
That's
But
absolutely right.
I've always argued that when you did 4 weeks,
Yeah.
you really had, it was because it makes it convenient for people to remember to come back. Because if you say to me, come back in 6 weeks, and the numbers have significantly decreased,
Yeah.
let's say here, okay, in New York, there's some areas in New York, it's like 10% positivity. Okay. So whatever, let's say the whole state overall has 5% positivity and I don't know, 4,500 cases. Okay. And then in September, when and now I'm scheduled for my boost in September, and there are 30 cases, and it's not in the news or headlines anymore. Am I
Mm-hmm.
going to remember to go back? Am I going to feel
Yeah.
that it's necessary to go back,
No,
or it's critical to go back? I'm going to say, ugh,
yeah.
don't
Yeah.
need to go back.
That's
I don't
right.
want to go to the doctor. It's like
Melissa
going to the dentist.
says there's no data. There's an idea that people have disastrous long-term effects because normally the process to create a vaccine takes decades. Well, not decades, but 5 to 8 years fastest. And yeah, but we had to accelerate this, and now we know we can actually get a vaccine out in less than a year. There haven't been any adverse events that are life-threatening. So I would say, these disastrous long-term effects like Guillain-Barré are pretty rare. It only happened in 1976, so that would be my logic.
Yeah, I'm fine
Uh,
with that.
Patricia said a coworker got her first Moderna. Her lips and tongue tingled for about an hour. She has an allergy to wasp stings. Now uncertain if she should get the booster. Have your EpiPen. That's why they have you wait for 15 minutes after the inoculation to make sure you don't have an anaphylactic reaction. If you do, they'll take care of it. If her doctor says it's okay, in these cases, people with allergies, when you go, I guess they ask you, Amy, do you have an allergy to a vaccine? They don't ask you about any other allergies, right? Do you remember?
No, when I filled out the form, it just said, did I have any allergies?
Allergies, period. Okay.
Yeah. Um, and when I filled out the form for Ira, it asks me if he has any allergies.
I think you need to consult a physician.
Yeah, I don't like to answer those questions. I'm not a physician, I think.
Could ivermectin's overuse cause ACE2 mutation that will make vaccines ineffective? No, not at all. ACE2 is it's not going to cause a
Well, I don't
mutation
know
in
that. I
us.
was going to say, like, I don't know how if ivermectin would cause a genomic mutation. It's a pump inhibitor.
Yeah.
It's not nicking the DNA. It's not binding the transcription factor in a different place or something. So I don't understand.
Is thought being given to all the paraphernalia that's needed to deliver vaccines worldwide? Will there be enough? Yeah, for sure, because you can have all the vaccine you want, and if you can't deliver it, so people are, the logistics people, which is not me, and I don't think it's Amy either, right?
No, it's definitely not me. I mean, Alan and I had this conversation, right, in October, where he talked about, where we did some paper about polio, right? They were talking about, like, you know, getting the resurgence of VAP or something in Nigeria or something. Remember, we talked about Raul's paper, and you
Yeah.
did the polio update and something or other. And Alan talked about like why they use OPV versus IPV and getting needles all over the place and blah 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 and the infrastructure. And then my comment was, well, the whole point of this was to build the infrastructure.
That's right. And Miguel wants to know any pulmonary effects after vaccination, like what happened in mice with SARS vaccine. Apparently, there was ADE with a SARS test in mice. That was our friend Stanley Perlman, I believe. There's
Yeah,
no.
well,
Go ahead.
No, go ahead.
I don't know any evidence that this is happening in people. None of the trials showed any lung issues whatsoever. There were no such side effects reported. So I don't think the mice results were relevant to this.
No,
We
I think I saw a paper also that talked about that the animal studies were not relevant. And
Jamie
no, I
said
was falling off the chair. It's on wheels. I fell off.
there have been no serious side effects in the vaccine trial. They got vaccinated 6 to 8 months ago. Yeah, I agree. There have not been a lot. So far, the vaccines have been proven effective against disease, but not necessarily infection. If they don't prevent infection, how do we reach herd immunity? It's hard. I think they probably will prevent transmission to some extent. And I think there's some data already from Israel, right, and from the UK. So
Mm-hmm.
I think they will knock down transmission and that you'll be able to get some kind of herd immunity.
I think you can get herd immunity even if you don't knock down infection.
Transmission, you mean?
Yeah.
How can you herd immunity? You're protected by the immune people because they don't transmit to you.
No, you're just protected.
Why would you be protected if people are transmitting after being immunized?
Because so few of them are actually, you really don't have a lot of transmission. I think like actually you get herd immunity against measles, and I think it's not against infection. I don't think that vaccine prevents infection. And you would say that you get immunity against you get herd immunity against measles.
The disease. Okay.
Mm-hmm. And when you don't, because when you stop seeing, when you stop vaccinating, you see outbreaks almost immediately. That's what happened in Disneyland.
Yeah. Jason says, why aren't data available from phase 1 and 2 regarding longevity? It hasn't been long enough. Phase 1 and 2, I know, were earlier last year.
I didn't think phase 1 addressed longevity. I thought it just addressed safety.
Well, it's safety, but they do collect sera and look at, immune responses. So I don't know if they retained those people and went back. I don't know if that's part of as Amy says, longevity was not part of that trial design, and maybe not even phase 2.
Yeah, I don't know. I don't work for a company. I don't know.
Tomas says, Amy, aren't you worried about Pfizer and Moderna vax got emergency approval? The manufacturers are not liable.
Aren't I worried? No.
Because
I
he says
mean,
they're not liable if you have a problem.
I'm not really a lawyer. I'm not, you had to go through certain hoops, and the hoops were the same for everybody and for every vaccine that goes through an EUA and stuff. So it's not like they bent the rules, and they had, so I'm not really worried.
Kang wants to know if the same variation can arise independently in 2 or more geographically separated areas. So,
Yes.
but if you look at the whole genome sequence, there will be SNPs that allow you to distinguish where it is, right?
Yeah, but there are also hotspots.
Yeah. So you could have a spike change in the same place, but then the rest of the genome would say it's not the virus that came from that place. It arose independently.
Well, it also depends on what you're looking for. Right? We just finished discussing about the genetic code. So if you're just looking at changes, variation in amino acid, that doesn't necessarily tell you a lot about the genetic code, right?
Yep. Kara says Scotland has lower levels than from before B117. Any thoughts on why the variant hasn't caused higher infection rates? Well, Kara, Amy and I were never fans of the idea that this lineage caused greater transmission. And so, as it's gone to other places in the U.S., for example, it has not caused greater transmission. So I don't think it's more transmission. It may be in certain populations it's more fit. Maybe there are other viruses that outcompete it. I think it's a nuance, and it's not as simple as saying this virus is more transmissible.
Maybe it has to do with human behavior.
Could be. John Mito said, trial participants since last summer for Pfizer are the longest term data available. So far, nothing worthy of note. The vaccine trials are not over. That's absolutely right. They go on for another 2 years.
Right. But the other thing is you do have to agree that there were 30,000 people and they saw no anaphylaxis response, right? And when you started vaccinating millions of people, you saw it.
Mm-hmm.
So millions of people are on week number 8. 30,000 are on 6 months.
Mm-hmm.
You always say there's nothing like a million children's guts.
That's right. Well, it's in millions of people now. We'll see. If you're worried, just wait. You have that right. Eileen says, is there something distinctive about AstraZeneca, given that today's Times wrote it's known to decrease transmission? Don't all vaccines do that?
No.
All right. First, Eileen, no. All vaccines do not decrease transmission. The polio vaccines do not, at least the inactivated vaccine. And I would be careful about believing what you read in the New York Times. They're not always correct. Would you agree, Amy? They're not always correct?
Yes, they're not. Most mainstream media is not always correct. It cherry picks what it wants to sell you. I mean, there's many times my mother calls and she says, blah, blah, blah. And I say, no, 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 no. And she says, but that's what I heard on the news. That's what they said on CBS or NBC News or in the Times or something or other. And I'm like, yeah, but Mom, when you put the whole story, blah, 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 plus X, Y, and Z, multiply it by this and divide by 4, you get something different. And she's like, well, why don't they just tell you that? I said, I don't know.
Yeah, they're selling newspapers. MK says HPV vaccine is better than getting HPV for sure.
Yeah, but I don't think the immunity is different. Right?
Yeah, the HPV vaccine immunity is really good.
It is, but so is HPV immunity.
Yes. No, the immunity is different, but with HPV infection, you can get cervical and penile cancer, right?
Well, yeah, and then you get the warts, and they can come back, right?
Yes, yeah. All right, El Denny says, I heard something on Twiv, getting both shots in the same arm versus one dose in each arm. Is one better than the other for maximum efficacy? Do you know the? I don't know the answer, but this is what Daniel said. Do you remember what Daniel said?
No.
He said it doesn't matter. He said if you get in one arm, your local lymph nodes are going to make immune cells, and then they're going to diffuse throughout your body. So if you get the next shot in the other arm, they're going to be memory cells there anyway. So it doesn't matter if you get it in a different arm.
Right. But when you go.
Who, me?
Yeah, when you go, or at least when I went, they asked, are you right-handed or are you left-handed?
Yeah, they didn't ask me that.
Well, they asked me.
I said I want it in my right arm. And they said, oh, okay. And why?
Why would you want it in your right arm?
I don't know. No idea.
Well, I wanted it in the left arm because I'm right-handed.
She was standing on my left side, and I wanted to give her a hard time, I guess.
Apparently. But the thing is your arm gets sore. And so I do a lot of work. I can't have my right arm not workable.
That's fine. Mine was sore, but it still worked.
Well, mine was really sore, and it wasn't working as well.
Okay, here's a different way to phrase the question. How frequent are long-term effects in vaccines? So, again, Guillain-Barré is long-term. Can be long-term. Not always. Sometimes it reverses. So that is a long-term effect. 400 people, 1976. The only long-term effects in other vaccines I'm aware of that are known to be caused by the vaccine are the Sabin attenuated polio vaccines can paralyze about 1.5 million kids who get them. That's a long-term effect, and nobody talks about that. That's the one you should worry about. Unfortunately, we're phasing it out globally.
Right. I can take 2 more questions and then I have to add overlay to my plaque essays.
Donald wants to know, are the mRNA vaccines possibly safer than inoculation with whole virus? You mean whole inactivated virus or attenuated virus? But yes, the mRNA, it's one protein, so it's never going to give you the disease, right?
Right.
But yes, it has fewer epitopes. And Amy will say it doesn't have a lot of the other proteins, and maybe they make a difference, but we haven't looked. Right, Amy?
Yeah. I get the whole point of it, but I'm about the fact that a virus, usually the genome is pretty small. They don't have a lot of bells and whistles that aren't necessary for some reason. So just because from our one assay, it didn't seem to have an effect, or we can't figure out what the function is. It clearly has to have a function. Otherwise, it would have been lost. Do you know what I'm saying?
Yeah, I got it.
So, are you going to disagree?
No.
So, just because we don't know what envelope and membrane protein do, or they're important because when we delete them, maybe they don't have a replication defect in tissue culture. You really don't have any idea what they do.
No.
It's too hard to figure out.
All right, one more.
One more.
Seems like natural immunity is wearing off after 6 months and the trial participants still have antibodies. I don't think that's a correct statement. Antibodies wane always after infection. The question is if you have memory B cells and T cells.
Well, not only that, but there are papers by Cromer and others that talk about going out 8 months.
After natural infection, yeah.
Mm-hmm. That's what he does.
Yep.
He doesn't do, well, maybe now they're doing vaccines. I don't know. I didn't read this new paper yet. But he has other papers that talk about, it's been a complaint of mine.
All right, Amy.
All right, I gotta go.
Thank you very much.
All right.
Bye.
I will talk to you tomorrow. Bye.
All right, it's me alone. Let me just, nope, that's the wrong thing. VR alone. Where is that logo? Oh, thank you. I think this is from last time. Okay. A number of people are saying that some vaccines give better immunity than infection. I would, have to look at those data. I think you need to be really careful. Abraham says, what do you think of Jacques Pepin, whose book I really like, The Origin of AIDS, revising the origins to World War I? Well, I always thought it was, 1900 to 1930 with, 1920-ish. So I think the genomics have always said that. And so and maybe he's just coming around to it. Do vaccines in general affect the result of antibody testing? Oh, okay. So it depends what you test for. So the Pfizer, Moderna, they're spike vaccines. So you're going to make antibodies to spike. So if you look for antibodies to N protein, which is a different viral protein, you'll never see them because there's no N protein. So you have to look for antibodies to spike. You can get both antibody tests for spike or N protein. Evan also says, I wonder if the current vaccines to spike are trying to induce more of an IgA response. Some of them do. So I believe there have been some studies where they look for secretory IgA. And, of course, those would be great for preventing infection, and they do. But I'm not sure the levels, what that means. Russia is sending Sputnik vaccine to countries that cannot get or afford it. to Western vaccines. So now the latest paper just came out. We're going to cover it on TWIV on Friday on the Sputnik vaccine, and the numbers look good. The efficacy is good. So, yeah, I think it's fine. It's 2 adenoviruses. We know adenoviruses are okay. The AstraZeneca is a single adenovirus. The Sputnik is 2, so they don't interfere with each other. I think that's a great idea. And so I would, I saw the data. They look fine to me. There are no side effects, and it's effective. So I think it's okay. Explain the difference between new variants and new strains. So a strain would be a virus isolate that has some new property that you haven't seen before. And so, these variants, the UK, the South Africa, the South American, and others, they have a puzzling epidemiological pattern, but no one has figured out biologically what it's doing. And so until we do, we don't call it a strain. All right. So that's the difference. We call them variants. So Hubert wants to know, is it possible that the virus escaped from a lab? I don't think so. If you look at the sequence, this is that and a bad virus from 2013 have a common ancestor. And there are thousands of changes between SARS-CoV-2 and the 2013 bad virus. So one of the arguments that many people make is the Wuhan lab was working on the 2013 bad virus and it got out. It's impossible. There's no way that virus is SARS-CoV-2. It would have taken 10 years for it to mutate to become SARS-CoV-2. And you couldn't even accelerate that in the lab. And Wuhan, that's what they do in Wuhan Virology Institute. They study bat coronas and their potential to infect humans. They didn't have anything. They didn't even have that bad virus in the lab, by the way. They had a piece of its RNA, and they didn't even grow the virus at any point. So these arguments are just looking to blame someone, but they're not tenable scientifically. RE infection, vaccination, lasting immunity issues. What if consequences of natural infection is suppression of immune response? Yeah, then that's a problem, right? So, well, maybe that's why the measles vaccine is better, because wild-type measles wipes out your immune memory. So that could be part of it. Yeah, it's a good point. All right, so Plinky, she asked originally, how many mutations would lead to a phenotypic change? And then she says, what are the implications? That's a great question. So far, I see 2 implications for the variants that have been arising. One is that the spike changes may make vaccine-induced immunity less effective. And that's being tested a lot because that's important. And so far, it looks like it's okay. The vaccines will protect against the variants. And the other is whether these changes make the virus more fit so that it spreads better through the population. I think that's what happened in the UK. I don't think it was transmission. I think the virus just grew better and it established itself better in people. And that same fitness doesn't apply to people in California. That virus has been there since November. The UK variant has been in the US and in California since November, and it has not displaced the other strains. So it is not simply more transmissible. So what the changes are doing, we don't know. People don't know, and those are experiments that need to be done in the lab, and they simply haven't been done yet. All right, so Amy's gone. So I'll take some of her questions. If you had no antibodies but aren't memory cells, what you need to look for? Yes. It doesn't matter if antibodies go down after an infection, as long as they don't go to 0. If they stay low, that means you have memory cells that are keeping the levels at a low level, and that's what happens in infections and with vaccines. TWIV 714 is described that after FP insertion, fusion peptide insertion, class 1 fusion peptides turn before fusion. Has this been documented with microscopy? No, it's actually, some people have documented this with microscopy. Yes, the paramyxovirus field, that's been done. And I know that because the person on my floor at Columbia, she's got pictures on the wall. She's done that in her laboratory. But it's before that, and in most cases it is an extrapolation from pre and post fusion structure. That's right. What about waiting longer between first and second dose? Pfizer trials showed 91% protection. I think that's not the right number. It's not 91%. Or maybe it is. But anyway, 7 days. They didn't look after 7 days. We were just discussing this on TWIV yesterday. They only look for 7 days, so you don't know how long it lasted, right? I think you could wait longer because you need more time for memory and affinity maturation to occur after that first shot. I think 3 to 4 weeks is kind of pushing it. But as we've said, it's to get the trials done. But you could go in the UK 12 weeks. I think that's fine. I think it's absolutely fine. I go for a tetanus booster every 10 years. So there's no fixed time. Are CT values correlated with viral load, and is it correlated with shedding? All right, so the CT value is a measure of how much RNA. A low CT, like 10 or 11, is a lot of RNA. You need fewer cycles to get a signal in the PCR reaction. High CT value, 35, and so there's very little RNA. And shedding infectious virus, yes, we know there's a range of CT values where you shed infectious virus because that's been studied in people. And it varies depending on the persons and the machines and the primers. But, mid to high 30s, you're not shedding anymore. Is an asymptomatic infection possible with high viral load? I've seen some low CT values in asymptomatics, yeah. And that's why they transmit well as well. If I have been infected with different variants multiple times, do I need a vaccine? I'm a coronavirus magnet. I would say yes, because we don't know what your immunity is, right? We would have to test you, and we're not going to do that. So we're going to, yes, you should get the vaccine. And if we change it every year in the next years, right, because variants arise, like for flu, they arise, and we have to change the vaccine, you should get it. Yeah. Some of these questions I answered. Is it possible for a variant like the South African to mutate in two different populations and show up as the same variant when sequenced? I think that would be very unlikely, because if you sequence it, you're going to see the whole genome, and you're going to see differences elsewhere. Like you'll see the cluster of changes in the spike, right? You'll see it in South Africa and elsewhere. But then you'll see other changes that distinguish the two viruses. Now, you can always track the origin. You can say, this virus in California came from the UK or came from South Africa. You can always do that. Certain changes can arise independently for sure, but you will never have the exact same genome sequence arise independently in two places. There are just too many mutations that occur during replication. So someone asked, is there enough research available to say that they're more infective or worse? No, there is none. I explained it before. I'm sorry. And you may have heard it, but the comments and lag me what I'm saying all the time. In some populations, they seem to move through the population extensively. Like in the UK, B1117 moved through very quickly and displaced the existing strains. But then it went to California and it did not. So it's not a matter of simply transmitting better from person to person. There's just no way you can say that. It's a fitness difference most likely, but it's also population dependent. It's dependent on whether the viruses are circulating at the same time. And in this vein, whatever my thoughts on variants, should we be worried? No, Osterholm is a fearmongerer for sure, because he thinks that by scaring people, he'll get things done, the government to do things and so forth. So the existing vaccines are handling the UK, the South African variants as far as we can tell. Nevertheless, it's not going to make it worse in the next 6 to 8 weeks. Absolutely not. The Moderna is making a South Africa vaccine, for example, just in case we need to use that. And it's really easy to do this, make a new one and turn it around in 6 weeks. So I'm not worried. I don't know why her mic is louder. I have a meter here, and they both go up the same. Hello, Japan. Okay, a lot of these questions are already covered. Maybe I should go right to the bottom. My grandma, who has never had any vaccination due to old world superstitions. Would COVID be too intense? Well, if you have allergies, you might have an issue, but, I don't think so. I heard, who is it today? Someone emailed me. Her 100-year-old mom, who's in great health, got the vaccine and she's fine. Of course, that's anecdotal, but. Oh, this is a great question. Where do you think the hundreds of millions of syringes that we will use for COVID vax finally end up? So in our institution, they're incinerated, vaporized. Maybe not everywhere, but that's what we do. If you have strange neurological symptoms, should we seek a neurologist? Well, first I would go to a general medical doctor, an internist, who can then say, I know what this is, or, yeah, you should see a neurologist. That's usually the best way to do it. So this is a great question. Why is COVID rampant if these other viruses have all gone away? That's a great question. I don't know the answer. And it may have something to do with zero population immunity to the new virus, right? And the other viruses, there is some population immunity, and that together with masking and distancing, boom, it's gone. But COVID, there are a lot of susceptible people. And that's good because it means when we get to a certain level of population immunity, we should be better off. So did I hear correctly that having infection gives better immunity than the vaccine? It depends which virus we're talking about. We've had a lot of people commenting on how some are natural immunity is better and vice versa. So it really depends on the virus. Of all the COVID vaccines, only AstraZeneca uses post-fusion. What's the effect? Hard to say because their efficacy is 70%-ish, if I remember correctly. And I don't know if that's due to using post-fusion or not. I mean, the one advantage of pre-fusion, according to Jason McClellan on Twiv Friday, is that you can make a lot more protein. So, of course, in the AstraZeneca, it's an adenovirus vector that's delivering it. So maybe that's part of it. Maybe the virus made by the vector, there's just less of it because it's less stable. I don't know. Do I know why AstraZeneca is trialing in the U.S. two full doses? I don't know. I think they should have tried a half full because that worked really well in the UK. And I don't know. I have no insight. I don't know anybody. And Daniel Griffin doesn't know either. Okay, here's a common anti-vaccine sentiment. When some people die after getting COVID vaccine, yeah, do you know what they die of? Car accidents, heart attacks, strokes, and the people in the control arm also die. It has nothing to do with the vaccine. Doug says, further to earlier, is nucleocapsid in intact virions visible to antibodies? No, it is not. It's on the inside of the capsid, so the antibodies would not be able to attach to nucleocapsid in the virus particle. Now, here's the thing, though. Some antibodies can get into cells, and they may be able to attach to nucleocapsid inside the cell and do something. They may mitigate infection. We don't know, but the reason I'm saying this is for adenoviruses, we know that some antibodies can actually get in the cell and inactivate infectivity inside of the cell, so they don't actually have to bind the virus particle. So I would like you to take that thinking out of your repertoire. Don't think that you have to bind the particle to stop infection. The antibody can also get in and inhibit infection inside the cell. Okay, this is interesting. And TWIV 714 McClellan said Pfizer, Moderna, et cetera, use prefusion stabilized, right? And AstraZeneca uses not stabilized. Is that a good reason for not mixing vaccines? I don't think that would impact it at all. I think the spike is the key, and there are epitopes common to both. And also the AstraZeneca, there's going to be a fraction that are still in the prefusion conformation, so I think it's fine. Why aren't the mRNA vaccines given nasally? Well, they weren't tested. So you say, why didn't they test them? And so Moderna had been working on mRNA vaccines for many years. So I suspect they had data, which in animals, at least in mice, that suggested that intranasal didn't work. Okay, because if they had, they would have tried it, and they did not. I wouldn't be surprised if they're going to try it. It just may be that you just don't get the same uptake of the nanoparticles in the nasal mucosa like you do in a muscle. Production fail. Audio first, people. Audio first. Hey, Brett, goodbye. On my levels, it's the same. It's your end. Just fix it. Okay, vaccine hesitancy comes from deeper, crazier thinking than the 70s. My levels are really high on my end, folks. And just give me the thumbs down. Go ahead. Oh, God. No, flu vaccine was not contaminated with Campylobacter. There was a flu antigen that mimicked Campylobacter because Campy can do Guillain-Barré, but it did not have Campy in it. Do we have to get the vaccine each year? We might. It depends on what happens with the variants, and we'll see. But I don't think that's the end of the world because we do that with influenza, right? It's not so convenient, but I don't think that's a big issue. Yeah, so, and many people are saying this. Is it possible we need repeat shots? Maybe. I'm going to go to the bottom of the list here because people are complaining about me versus Amy. The audio has been fine. Thank you, Bill. Is the virus bigger than salt? No. You can fit several hundred viruses on the head of a pin. Yes, and we're talking about the sound is great. Thank you. Some people, they get cranky and then they yell at me and they give a thumbs down. Do only mammals have ACE2 receptor? What a good question. I don't remember this. That paper in PNAS a while ago that looked at them. I don't believe that that's the case. And I think the ones not in mammals are very distant, so the virus will not bind to them. Even many mammalian ACE2s won't bind SARS-CoV-2. Sound was great tonight. Thank you very much. I feel better now. I felt sad that my sound was no good. Oh, this is interesting. Has there been any research done with people who have Delta 32 and COVID? So Delta 32, of course, CCR5 gene deletion and makes you resistant to HIV? No, not that I know of. Get the vaccine every year. I'm still trying to get my 92-year-old mother her first vaccine. Yeah, I know. It's a pain, isn't it? I'm just saying, though, if we had to do it every year, it wouldn't be the end of the world. Because a lot of people are saying, oh, my gosh, the variants are going to kill us all. It's not the case. Immunoassay after vaccination. Daniel Griffin says don't do it. I don't see why you can't. Here's the thing. If you get a spike vaccine, you need an immunoassay that measures spike antibodies. Some of them measure nucleocapsid antibodies, so you don't want to get those, okay? Can viral infections cause Guillain-Barré? Yes, but I haven't seen any Guillain-Barré associated with COVID-19. But you're absolutely right. Not just vaccines that cause Guillain-Barré natural infection. Some people develop Guillain-Barré without even knowing what causes it. So Albert Sabin, who developed one of the polio vaccines, one year when he was in his 80s, suddenly developed Guillain-Barré for a few months, which is a paralysis, and then it went away. We have no idea what happened to him. All right, Barack wants to know how antibodies can get inside a cell and how can this suppress infection. So things get in cells by endocytosis, right? And antibodies can get into cells by endocytosis. Sometimes they get in with a virus particle. In the case of adenovirus, they're taken up, and then in the cytosol, the antibody and the particle get out, and then the antibody can attach to some parts of the particle that are normally not exposed outside of the cell. So cells take up a lot of things. That's how antibodies can get in. I'm sorry, Nicoletta. You were called condescending when you said to search academic papers instead of the Times. Well, that's the right thing to do if you want the real answers, because look what's happening in the Times. The science writers are calling reports. So they get a press release, and they look at it, and then they call experts, and they talk to them, and then they paraphrase what they're saying. So it's all many times removed. Now, I'm not trying to diss science writers. There's some very good ones out there. But if you really want to know about viruses, who do you think you should talk to? And it's not arrogant or condescending. Talk to a virologist. Yeah, so Israel now, there's a preprint out from Israel where they think they have changed the course of the outbreak by a week. And this is by a combination of data and modeling. So it is not clear that to me that is the vaccine. But we'll see as time goes on. They've done a very good job. They've done a great job. Do you think Merck will help produce one of the other mRNA? So Merck is out of the vaccine business. And Sanofi, as far as I know, is not doing anything at the moment. Can we speak at broadly acting antivirals in the works? So, yeah, we will. We talked about one a couple of weeks ago. It's called NHC, and it was a study in ferrets. And this is a drug that inhibits a lot of RNA viruses. And it inhibits SARS-CoV-2 transmission. And you know what? It's been around for a couple of years, so it could have been tested a long time ago. It's unfortunate. When will SARS-CoV-2 stop mutating? Never. It mutates all the time, in every person, in every cell. Now, here's the thing. These variants that are arising, they have like 15, 17, 18 amino acid changes in the genome, which is more than we have seen up until the end of last year. And I don't know why they're more. People are speculating that these are coming out of people who have long-term virus replication. I don't know if that's the case. But as long as there are people infected, and that's part of the problem. So many people are infected that lots of variants are being generated. And if you get one that randomly is just a little better, it might take off. So that's the issue. Has there been any interaction with vaccine and daily drugs? I have heard of none. According to Daniel Griffin, there's none that he has flagged. So take that. Thank you, Questella. I appreciate it. I really appreciate the thumbs up, folks. I don't know the answer. Autoimmunity, anti-nucleic acid antibodies would be a theoretical immune impact if you had COVID. So I don't know the answer, but here's the thing. The long COVID is thought to involve some autoimmunity. So a person who's already has autoimmunity issues, that might exacerbate it, right? So I don't know the answer, but it seems to me a possibility. Draconic says I've been Racanielling. Sorry. Yes, maybe it was John Campbell. People like to complain, right? But most of you are fine. You don't complain about the sound and so forth. Although, remember the first live stream, there was an echo, and I fixed it. And I knew that was true because you were all saying it. CDC is funding 6,000 genomic tests per week. Is that enough? Too little? I don't know what the right number is. Somebody asked me that the other day. Whatever it is it has to be more than we were doing. And so 6,000 a week, that's 6,000 isolates. Here's the key. It's got to be spread around the major population centers to pick up variants, and then I think it's okay. Did you see the H3N2, H1N2 variant found in Brazil? Are you sure H1N2? I've never heard of a human. It may not be human then, because H1N2 is not a human virus. And I didn't see any change of the vaccine for that. There is a change, but it doesn't include that one. Is there less influenza virus in the community, or are our public health measures causing low prevalence? I think it's public health, and there's quite a bit of population immunity to influenza. And as I said earlier, that probably combines with masking and distancing to really make an impact on the outbreak, right? Can a single person be infected with different variants? Yes, actually, you could be. And coronaviruses are very good at recombining. So you could get new viruses, and maybe that's how some of these variants arose. We really don't know. In terms of disease, I don't think you would notice the difference. But, some variants may emerge that are more fit, and they could spread on. Good night. A lot of comments that I don't need to answer, folks. Sorry about that. Thomas, it's not a mistake. There's no proof that these are more transmissible. If you listen to me when I say transmission is going from person to person, there's no proof that this has a better person-to-person spread. It may be spreading through the population, but Thomas, how do you explain that the same variant goes from the UK to California and does not spread through the population? It cannot be biologically more transmissible. So don't tell me about mistakes. That's the facts. It's not about mistakes. Should we reopen the economy once we have vaccinated all over 60s and vulnerable? Isn't it open already? You mean going to restaurants and all that? I think we have to see the impact of vaccination over the next weeks. And if infection numbers go down, then we can start to think about it. In New York State, Cuomo said in March you could have gatherings of 150 people or more. So people in Russia who get vaccinated get tested for anti-spike. Everyone knows their titers. Yeah, so you heard Daniel. He was against the idea. So what do they do with the titer information? Do they do anything with it? That's interesting. Danny, I'm sorry if I miss your questions. The chat goes by pretty quickly here. I'm doing my best. Is AstraZeneca sterilizing? No, it is not. The latest numbers suggest there's some impact on infection, but it's not 100%, which would be sterilizing. How do I feel about YouTube censoring medical doctors? I don't know anything about it, frankly. If I could design a vaccine against SARS-CoV-2, what antigen would I target? So that's kind of a hard question to answer. Because we already have these pretty successful spike vaccines. Now, when they first started developing these spike vaccines, I thought it was a mistake to focus on just spike. So what could be better? So I would love to include other viral proteins like nucleoprotein, and maybe you would get T-cell epitopes against those that are useful. And you may ask why. So here's the thing. the T cells will recognize virus-infected cells because they'll display the nucleoprotein on their surface by MHC molecules. And so those infected cells can be killed by T cells. So that may contribute to clearing an infection. Overjoyed, thank you. ACE2 is in vertebrates, including birds, fish, and amphibians. So more than mammals, right? And I'm sorry if I miss your questions. Don't get insulted. And if you don't like that, don't give me a thumbs down because you don't like it. That's just juvenile, don't you think? And Thomas is back saying, I don't know anything about epidemiology. I'm not talking about epidemiology. I'm talking about the biology of the virus. There's no data that tells me that this biology has changed in a way that makes it more transmissible. It's spreading in the population. And don't tell me what I can talk about. I've been working on viruses for 40 years. Have you? So you have a chip, you have a thing to contend because I don't agree with you, but grow up. That's the way science is. Maurice 2, thank you. If you do not have the expertise to understand it, the times will, on average, be more informative. Okay, but what if they say things that are wrong? For example, the what was it? The Novavax vaccine didn't do well in AIDS patients in South Africa, did 50%, but in the non-AIDS population, it did 60%. So what did the Times put in the headline? Only 50%, which is totally disingenuous in my opinion. But I agree it's better than most. But you know what, Victor, that's why we do TWIV. That's why I do all this SciComm. I'm a scientist who can explain things to people. I can't reach everyone, but not many people listen to TWIV. Not many people watch the YouTubes. It's too complicated. When I add super chats, I don't want to be seen as selfish. We have people donating, but I could add a super chat, see how it works. Thank you. I've seen that on other chats, of course. Is there any reason to think the evolution of the virus will be towards milder and more transmissible variants? Okay, so this is a thing I think about all the time. So I'm very interested from a biological viewpoint, not epidemiology, because I will admit I'm not an epidemiologist, but that's not all you need to understand SARS-CoV-2. Where was I going with that? I'm really interested in how these properties evolve because theoretically, viruses, and you might have listened to one of my lectures because I say this all the time. In theory, viruses should evolve to be less pathogenic and more transmissible, right? So they can infect the most number of people and cause less disease because less disease, more transmission. However, that is not what we see. We do not see that. And so we see viruses who maintain pathogenesis for many, many years, and it may be that helps transmission. So the best example is the infection of rabbits in Australia with a pox virus to try and control the rabbits. The first year, 99.9% of the rabbits died, and the 1.1% that lived after that were then more resistant to infection. So the virus became less virulent. But in people, we have very little data. That's why if you tell me in this outbreak there's a more transmissible virus, you better provide proof because I'm thinking from a theoretical basis that can a virus become more transmissible? And I have never seen a human virus become more transmissible during an outbreak. So if this is the first time, I want to know it, and I haven't seen it yet. And you shouldn't say I'm wrong because the data are not there. So there are some reports that ivermectin can help COVID, and Daniel Griffin has talked about that. And this is not just an anti-worm disease, but it can take care of other infections, and that may be what's happening. It doesn't seem, in my opinion, it doesn't have antiviral effect. It doesn't really affect viral loads in these patients. So it's doing something else positive, but we don't know what it is. People say that more strong reactions after the second dose. Could it be we are giving it too soon? I don't know what you mean by letting the immune system normalize, right? I think if you waited a couple of months, you would still have a strong memory reaction. So I don't think it's that. I think we should let the primary response mature a bit more, but we don't have the luxury of time, right? We want to get everybody immunized. What made the 1918 pandemic end without antibiotics? All right, so I asked an epidemiologist, okay? Why not? And that was Adam Kucharski, and he said herd immunity. Eventually, enough people got infected. But I think it was other things. The troop movements ended. I don't think you can ascribe it to one thing like herd immunity. What are the features of viruses? It's 9:25. 5 more minutes, folks. I don't want to keep you forever. Are there features of viruses that are predictive in terms of propensity for recombination? Yeah, there are some. So, for example, all right, so there's recombination, which is joining of two different sequences of two viruses, and then there's reassortment, right? So if you have a segmented genome, you reassort. Influenza, rotaviruses, many other viruses have segmented genomes. Then we have recombination. Negative strand viruses do not recombine, RNA viruses. We don't see recombination in them. So measles viruses and mumps, et cetera, they do not recombine. So that leaves us with the plus strand RNA viruses. And they're all pretty good at it. Polio, 10 to 20% of molecules in an infected cell are recombinants. The coronaviruses are really good at recombination because the way they make their mRNAs involves polymerases jumping from one template to another. So that's the recipe for recombination. So yes, there are certain things you can do to predict recombination. Would putting an ice pack on help? Yeah, you could do that if you wish. How long will it take for this virus to lose its potency, morphing into the common cold? So Ralph Barrick, and someone asked how Ralph is, and we might have him back on again, to see what's happening. He's working away. He's publishing a lot of papers. How long will it take for this virus to lose its potency? That was first suggested by Ralph Barrick back last year at the beginning of the outbreak. And he said, eventually when the whole population is immune, or much of it, this will not cause much disease, because the problem now is it's entering in a non-immune population and older people are getting sick. So he said, we don't know how long it would take. It could take 50 years. It could take 400 years. So the common cold coronas emerged many hundreds of years ago, and we don't know when we suspect that when they emerged, they were pathogenic and then they became mild like they are today, but we don't know the time frame. It's not going to happen in a year. Is any oral vaccine available for COVID? One is in development. There's an adenovirus vaccine, vectored vaccine, which is put in a capsule. It passes through your stomach, goes into your intestine, and it immunizes your intestinal mucosa. And then, your mucosal surfaces tend to get immunized at the same time. You immunize one, and then others get immunized. So that's going to give you respiratory immunity. But that's in still very early phase of testing. It's not available yet. Okay, we'll have Barrick back on TWIV. Can we have a Q&A with anti-vaxxers and virologists? You'd have to get the right anti-vaxxers, and I'm telling you, that's happened before, and they just never come around. They never change their view. So Andrew Wakefield, for example, Robert Kennedy, they just go around in circles saying you're wrong, you're wrong, you're wrong, and I'm not sure it's productive. You want me to go easy on mainstream media? Look, Tom, you have no idea how it bugs me when they get things wrong. So you're right. I agree that we have other issues with other news sources. But I go on a Fox radio show every week at Monday morning at 9:30, and that guy has never dissed me. He said he trusts me for his science. So And I didn't even know it was Fox when I first went on. I've been going every week for the last year. So I try. I do try. Thank you so much for your comments on the sound. Every country needs to be doing sequencing. Yes, but not every country can, right? They can barely take care of their people, so we have to help them. And we need to help them sequence. We need to give them vaccines and antivirals when they're available, but we don't have that. What am I going to say when Florida gets an outbreak? Then I'll say, yes, in Florida, the fitness has driven it through the population, but it clearly hasn't in California. Okay, one more, folks. Maria, I read in the press an article the virus could stay in brain cells and be reactive. So, no, I don't think it stays in brain cells. I haven't seen any evidence for that. I think most of the brain symptoms are caused by immune reactions against the infection. I'm not convinced that the virus reproduces in the brain, and therefore it's not going to be dormant there. All right, folks, thank you all so much for joining us tonight, Amy and myself. I really appreciate that you're all here, that you participate. I even appreciate the negative comments, and I know I shouldn't respond to some of them, but I do feel strongly about them. And we will be back next Wednesday at the same time and place. And so thanks for coming.